Hi, I'm Lauren Levy. Today I will be presenting the 2018 paper, Non-Invasive Delivery of an Alpha-Synuclein Gene Silencing Vector with MR-Guided Focused Ultrasound. This work represents collaborative effort between the Tandon and Aubert labs. So, background. As some of you may know, Parkinson's is a common age-related disorder that primarily affects the central nervous system. Initially described by James Parkinson in 1817 as the shaking palsy, it is primarily associated with motor symptoms such as resting tremor, rigidity, bradykinesia, and postural instability, though non-motor symptoms such as loss of olfaction, REM sleep disturbance, constipation and incontinence, and orthostatic hypotension have been implicated. Braak staging suggests that neuropathology of PD begins in the dorsal motor nucleus of the vagus nerve and the anterior olfactory nucleus, and later spreads to other regions, such as the hippocampus. Most notably, degeneration of the pigmented neurons in the pars compacta of the substantia nigra result in dysfunctional motor control. While many genes and pathways have been implicated in the pathology of PD, Lewy bodies, which begin with the aggregation of alpha-synuclein, remain a hallmark of the disease. Alpha-synuclein is considered both a cause and endpoint of Parkinson's disease. Under healthy conditions, the blood-brain barrier protects the central nervous system, giving it what is called immune privilege and selectively preventing solutes from passing from the blood to the extracellular fluid of the central nervous system. However, this protective mechanism complicates administration of therapeutic agents, which often cannot pass the barrier or have other drawbacks. The AAV9 serotype, for example, requires high doses and still fails to reach the entire CNS. A variety of strategies have been developed to overcome this, and in the past few years, a combination of magnetic resonance-guided focused ultrasound and phospholipid microbubbles has shown great success in locally, transiently increasing permeability of the barrier. When the microbubbles reach the targeted region, the ultrasound waves cause the bubbles to absorb the energy, expanding and contracting. This is called oscillation. As they expand, they stretch the capillary walls. This allows the viral vector to carrying either a short hairpin RNA to silence alpha-synuclein or a scrambled version of that sequence to cross the blood-brain barrier. Use of a scrambled vector is considered a standard control for testing viral vectors. Having set the stage, we move on to materials and methods. Novel methods practiced in this study include targeted focused ultrasound and the shRNA gene silencing, along with more common components and techniques such as transgenic mice, immunohistochemistry, confocal microscopy, image day quantification, and standard statistical analysis. As previously mentioned, crossing the blood-brain barrier is a typical therapeutic agent. One additional advantage of focused ultrasound is localized permeability, which enhances the precision of therapeutic agents. For this study, the hippocampus, the dorsal motor nucleus of the vagus nerve, the olfactory bulb, and the substantia nigra were all targeted separately by focused ultrasound, selectively enhancing the permeability of the barrier that insulates the vascularization supplying those regions. Short hairpin RNA, or shRNA, for those of you who do not know, is an artificial RNA molecule that enables targeted gene knockdown through RNA interference, binding messenger RNA and co-opting the RNA-induced silencing complex, or RISC, to either cleave or repress translation of the mRNA. In this case, the AAV expressing shRNA targeted nucleotides 299 to 309 in human alpha-synuclein, while the control group expressed a scrambled sequence. Additionally, to reduce cell death associated with human alpha-syn shRNA, both vectors included short miR30 microRNA insertions flanking the 5' and 3' ends of the silencing sequence. Both vectors included the cytomegalovirus promoter, the reporter turbo-GFP, and an internal ribosome entry site. Transgenic mice expressing human alpha-syn complemented the choice in viral vector. I asked Professor Tandon, one of the corresponding authors, about why this was. You can find a complete answer in the interview portion of this presentation, but the gist of it is that this was primarily a proof of principle study. Additionally, upregulation of beta-synuclein could rescue loss of alpha-syn. Immunohistochemistry was used for a couple of experiments. To determine the extent of alpha-syn knockdown, triple labeling for alpha-syn, synaptophysin, and tyrosine hydroxylase was performed for the hippocampus and substantia nigra, while labeling for alpha-synuclein, synaptophysin, and choline acetyltransferase was performed for the olfactory bulbs and dorsal motor nucleus. Synaptophysin is highly expressed in nerve terminals and was used to normalize the intensity of the alpha-synuclein immunofluorescence, while presence of choline acetyltransferase and tyrosine hydroxylase were indicative of cholinergic and dopaminergic cells, respectively. Tunnel, cleaved caspase-3, and neuronal nuclei, or NUN, were indicative of cell death, while IBA1 and GFAP were used to identify activation of microglia and astrocytes. Statistics were based on a paired two-tailed student's t-test. Okay, now time for the results. First, the authors had to confirm localized permeability using T1-weighted MRI to confirm a gadolinium-based contrast agent. Under normal BBB permeability, it would not penetrate, but focused ultrasound selectively enhanced local permeability. Additionally, the scrambled and human alpha-synuclein vectors demonstrated no significant difference in peak pressure and relative permeability in the target regions. The virus did not cross the blood-brain barrier without focused ultrasound. Next, they demonstrated that a month later, there was at least 60% knockdown of alpha-synuclein in the hippocampus, substantia nigra, and olfactory bulb, with approximately 35% knockdown in the dorsal motor nucleus. Synaptophysin expression remained similar between the two vectors, suggesting that synaptic integrity remained unaltered by focused ultrasound gene delivery and could be used to account for anatomical variations in alpha-synuclein levels. Incomplete gene knockdown is ideal because low alpha-synuclein has been associated with a decline in nigral tyrosine hydroxylase and possible neurotoxicity. However, there was no change in nigral tyrosine hydroxylase immunoreactivity. Finally, they checked for markers of cell death and inflammation. To identify apoptosis, they immunolabeled transferase-mediated DUTP, fluorescein-Nick end labeling, or tunnel, cleaved caspase-9, and neuronuclei, NUN. Tunnel was also evaluated with an in situ cell death detection kit. There was some evidence of cell death in the olfactory bulbs, which the authors attribute to normal granule cell turnover. It's visible in what seems to be similar levels in the non-focused ultrasound scrambled vector control, the focused ultrasound scrambled control, and the focused ultrasound alpha-synuclein treatment. That same apoptosis doesn't seem to be present in any of the other anatomical regions. The distribution and intensity of astrocyte and microglial markers, GFAP and IBA1 respectively, were similar. Previous studies have reported a transient increase in inflammation following focused ultrasound. While there is some risk involved, it is much safer than surgical or penetrating routes of administration, and distal routes don't allow for localized administration. So looking at the conclusions and future directions, this study was a proof of principle that represents the first of its kind in offering localized administration of a viral vector for gene silencing in multiple target regions. What this means is that there is no need for surgery. So on one hand, there is some risk of enhanced permeability, but that risk is significantly lower than performing brain surgery to inject it directly with a syringe or a needle. This also means that we can target multiple regions at the same time, which again isn't possible if we were to inject something into the tail vein of a mouse. So we have enhanced bioavailability, and minimal invasiveness. In the future, we might also see rescue of alpha-synuclein knockdown with upregulation or replacement using beta-synuclein, a highly homologous protein. The future additionally holds improved localization with more precise ultrasound and other strategies to selectively target pathological alpha-synuclein, such as nanobodies that could block aggregation and instead sequester alpha-synuclein, bringing it to, say, a lysosome. In the future, we expect that viral vectors, nanobodies, and other therapeutics will be improved over time, enhancing their precision and enhancing their accuracy. But what this study does uniquely is it establishes that gene silencing and gene knockdown can be linked with focused ultrasound. Focused ultrasound, again, will also likely improve over time as we enhance our ability to target certain regions, as we enhance our ability to identify the precise, frequency, and so on. There are factors that will continue to evolve, but what this does is establish focused ultrasound as a key component of future therapeutic administration. So, as I mentioned before, I did an interview with Dr. Tandon, and I am going to include it here.
My interview with Professor Anurag Tandon by Lauren Levy. So I have a couple questions today about your paper on non-invasive gene silencing for alpha-synuclein. My first question is about the blood-brain barrier. The blood-brain barrier under healthy conditions is a protective layer. I was curious if there's any risk of introducing toxic elements or debris to the brain when the blood-brain barrier permeability is enhanced by focused ultrasound and microbubbles, such as immunogenic antigens.
Yeah, that's actually one of the first questions most people ask, given we know we have a protective layer and we are disrupting it. Now, you just have to remember that in any kind of brain surgery, first of all, the first thing that happens as you cut into the brain, whether it's for epilepsy or cancer or anything else, you're always cutting to the blood-brain barrier. So the blood-brain barrier is always being slightly permeated by any type of neurosurgery. And the goal of our approach is to minimize the length of that time. So in this case, we know that in about 6 to 8 hours, the blood-brain barrier is closed. And this has been done repeatedly in animal studies and more recently in human trials, for example, for Alzheimer's disease, where, as you may know, there is also a risk of vascular amyloidosis in blood vessels of the brain. And so it was a real challenge. The question was to try and address whether those blood vessels in Alzheimer's patients would also react the same. And in fact, they did. In an early trial with 5 patients that had 3 repeated focused ultrasound treatments over several weeks, what they showed was, this was a study done at Sunnybrook with my colleagues. What was shown was that the brain was, the blood-brain barrier was opened, and when the patients came back the following day and were scanned again, it had completely closed over. And the way they do this is using a MRI tracer, which is injected in the bloodstream, which doesn't cross the blood-brain barrier under normal circumstances. And you can see it permeate when the blood-brain barrier is opened. And the day after the sonication is done, the opening is completely closed. Now, the question is related to whether there are, pathogens that might get crossover through an open blood-brain barrier, and that is true. So the risk of having viruses or bacteria that might be bloodborne that can pass into across the blood-brain barrier when it's permeated is not a trivial issue. And so, as in all surgeries, patients have to undergo blood tests and also be assessed for any sort of viral infections they might have. So those are the normal pre-surgery precautions that are taken under any type of surgery. Now, so far in the animal studies, it doesn't appear that they have the blood-brain barrier opening does not seem to induce any deleterious effects. The question is whether repeated openings might, but even there, it seems that as long as you don't open the blood-brain barrier to the extent where, excuse me, large blood cells are also crossing through, it seems to be quite safe. And in some cases, there have been a number of studies that have suggested that it might even be beneficial, for example, in terms of neurogenesis, in terms of improving how the brain reacts to various insults. So I think the long-term prognosis for blood-brain barrier opening, I think, is quite good. It has very limited number of deleterious side effects and a lot of positives in terms of not requiring direct insertion of surgical instruments or any other tools into the brain.
Wow, okay. So is actually, out of curiosity, why would it be beneficial? Like,
I'm
what could
sorry?
cause? You mentioned that it could be beneficial. Out of curiosity, like, why would it be helpful?
Yeah, we don't. I
Therapeutic.
think that's been shown mechanistically or empirically. We don't know what the rationale for it or what the basis for it is. We do know that there are increased number of new neurons that grow out as neurogenesis seems to be stimulated. There's also the possibility that certain types of factors peripherally may have a beneficial effect in the brain. And again, we don't know what that is. So for example, when you look at treatments of Alzheimer mice, and this is work done in Isabella Baer's lab, showed that when you sonicate animals that have high levels of amyloid in the brain or beta amyloid, you can also treat them with A-beta antibodies. Those antibodies tend to reduce the amount of A-beta accumulation. However, if you use just the sonication alone, there is still a significant benefit from sonication alone relative to the unsonicated hemisphere. And it's not as good as having a sonication and the antibody, but there is clearly a benefit, which suggests that there may even be anti-amyloid antibodies floating around peripherally that are delivered to the brain when the sonication is done.
Wow.
The short answer is we don't know what benefits there may be of opening up the blood-brain barrier. There certainly are risks involved, for example, as you mentioned, from pathogens. But as long as you control for those and make sure that the patient is healthy, it is very likely that those would be trivial issues when you're dealing with clinical delivery of drugs.
So exciting. Okay,
Yeah.
I'm going to move to the next question.
Okay.
So transgenic mice, I noticed that you chose to use transgenic mice with wild-type human alpha-synuclein rather than mutant alpha-synuclein. Is there a particular reason for that?
There was no need to use the mutant mice. For example, the A53T mice over time do develop some aggregation, but the mice we chose are lacking the mouse synuclein protein. So they've been knocked out for the murine synuclein gene, and they only express the human gene, which is a transgenic version. So for our purposes, the goal was to really knock down alpha-synuclein levels in general. And this is a proof of principle study where we could target specific parts of the brain. And so there wasn't any need to do any pathological analysis of aggregated synuclein. It was really to see whether the delivery of virus with anti-alpha-synuclein shRNA could knock down synuclein expression in specific parts of the brain. Now, there are a number of other ways to do this study, like you mentioned, using mutants, mutant alpha-synuclein. And one of the other studies that we have now underway is, in fact, looking at mice expressing alpha-synuclein A53T mutants. And the rationale for is simply that in this case, we want to rescue the pathology. Now, in the future, it's going to be important to not rely on mutations, because that's a very small part of the human population that actually has PD as a result of altered gene expression or mutations. So our goal in the long run is to be able to target healthy synuclein expression to see whether we can knock that down, because we expect that most idiopathic cases of Parkinson's disease are not related to overexpression or mutations. In fact, most of them are would appear to be normal alpha-synuclein that has been altered post-expression in some way. So some sort of post-translation modification and age-related changes that induce synuclein aggregation.
Wow. Okay. That's really cool. Question 3.
Okay.
Alpha-synuclein silencing in therapy. So alpha-synuclein is often associated with its potential for toxic gain-of-function aggregation. However, it is also associated with a variety of functions under healthy conditions, and loss of healthy function may also be a toxic pathological feature related to Parkinson's. So I understand that the vector used includes short MIR30 microRNA insertions that reduce toxicity in dopaminergic neurons relative to the shRNA lacking insertions, and that the gene silencing applied in this study was associated with an absence of cell death. Nonetheless, I'm curious, what role would knockdown or gene therapy like this play in therapeutics?
Yeah, that's a great question. And it's one that we don't currently have an answer for, because there's a lot of discussion in the literature that knockdown of synuclein might be partly toxic. There are several groups that have looked at changes in immune function, for example, or in the health of certain parts of the brain following synuclein knockdown. Some groups have found that it does seem to cause some toxicity in our hands, and other hands have not shown that. Now, the difference between some of these studies might be related to the extent of the knockdown. So our goal has usually been to reduce synuclein levels by about 50%, so that they still have a sufficient amount to undergo normal, let's say synuclein's involved in vesicle trafficking. Well, we would expect to see the same amount of vesicle trafficking even if it was knocked down by 50%. Those other studies that looked at very, very high knockdown levels of approaching 80 to 90% knockdown suggest that yes, synuclein, if you completely remove it, it will have a deleterious effect. Now, there is a way to get around this. We know if you think about the word alpha-synuclein, you might assume that there are betas and maybe gammas, and then you'd be right. So there's a beta-synuclein and a gamma-synuclein. The gamma-synuclein seems to be mostly associated with peripheral tissues. However, beta-synuclein has a very much brain-specific localization, and we think that it probably will compensate for synuclein knockdown effects if you were to increase it. So, and the reason we would use beta-synuclein also is that it's lacking the sequence that is involved in aggregation. So there is a core sequence in alpha-synuclein that self-assembles, and that is missing in the beta-synuclein. So one possibility would be, as we knock down alpha-synuclein, we would upregulate in the same vector beta-synuclein levels. And that might be one way of getting around the loss of synuclein function. And again, as I say, the loss of function of synuclein may be impactful not just in the brain, but also peripherally. There is some evidence that synuclein plays a role alpha-synuclein plays a role in innate immune function. And so there may be roles of alpha-synuclein that we don't fully appreciate. We tend to think of it as a brain protein, but it is expressed peripherally as well. So my goal would be to show that we can knock it down safely, but also in cases of some sort of toxicity, we would then try and replace it with some beta-synuclein or even a modified version of alpha-synuclein that doesn't self-assemble.
Wow, that's so cool. Okay, number 4.
All right.
So healthy alpha sign versus aggregate alpha sign. As a follow-up question to that regarding both gene silencing and antibody-based therapeutics, can therapeutics like this inhibit toxic alpha-synuclein while maintaining or restoring? I guess that would be resolved by upregulating beta-synuclein. But if so, I guess the considerations would be removing the portion that is involved in aggregation of alpha-synuclein.
Yeah, and that's also a terrific question. Now, the way we've done these studies right now has really been to deliver a viral vector that can knock down alpha-synuclein levels. But as you mentioned, the nanobody studies that we're starting soon would also allow us to target total levels of alpha-synuclein, but if you were to target an antibody, and I don't know if your group is aware of what antibodies are, these are simply very, very small components of antibodies that are composed of just the heavy chain, the variable domain of the heavy chain. So that's the part of the antibody that recognizes an antigen. We can clone these, put them into a viral vector, and then deliver them into the brain to either express intracellularly or extracellularly. And so we have one that can bind to intracellular alpha-synuclein and deliver it to the proteasome for protein degradation. The basis for this study is simply to look at whether we can knock down total synuclein. Now, at some point, we might be able to target only aggregated synuclein, for example. And so there may be another approach that we can make intracellular nanobodies that target only specific forms that we think are deleterious and remove those. And we could even get more clever about it and put on tags onto our nanobody that only go to specific cell compartments, for example. Let's say we want to target synuclein that accumulates in lysosomes. And we know with age, lysosomal function tends to be less robust. So we could target these nanobodies to be delivered directly to lysosomes, where they would bind up fibrils or oligomers of alpha-synuclein and then sequester them. They may not destroy them per se, but they would at least be able to sequester them and prevent them from self-assembly. So there are lots of ways to play around with this, and the challenge for us is to find one or more than one that works.
That is so cool.
Yeah.
Okay, I guess final question.
All right.
Just in general, what future directions or challenges do you foresee addressing next with regards to the use of focused ultrasound and alpha-synuclein in Parkinson's disease?
Yeah, so one of the main challenges for any drug delivery mechanism, and focused ultrasound is a particular one because when we're targeting the brain, one of the main aspects of why focused ultrasound is useful is because it allows us to target a specific part of the brain. Otherwise, you can just inject antibodies into the bloodstream. Maybe less than 0.5% will get at the brain. The problem is when you want to target a part of the brain, the question you can ask is, what part of the brain do you want to target? And so depending on the stage of the disease, from early to middle to late, your targeting may be very, very different. So very early in the disease, and that was one of the rationale for our study, where we targeted the dorsal motor nucleus, the substantia nigra, the hippocampus, the olfactory bulb, because these are all targeted either early or late stages. And so we know we can target them, and we know that we are able to deliver the vector to those particular areas. The problem is once you get into a clinical situation where you have a patient who presents with Parkinson's disease, how do you know which stage they're at? And so we may be required to do imaging studies. And one of the problems currently is that there are no good PET ligands for alpha-synuclein. There are ways to measure total dopamine storage. So you could do fluorodopa uptake. So there are ways to look at the pathology in a non-invasive way as well. And I think those things will get better over time. So the long-term goal for any Parkinson gene therapy or small molecule therapy and drug delivery will require us to know what stage the patient is at. Can we target that part of the brain effectively so that it prevents pathology from moving to another part of the brain? And I say this is a real technical issue because we don't think we can at the moment rescue cells once they've died. So if you've lost a part of your brain that is involved in a specific function, the neurons in that part of the brain are damaged, we don't have a way of rescuing those and bringing back healthy neurons. There may be a time where we are able to do that. So for example, you might be able to deliver a growth factor that allows a particular cell type to grow You might then have another factor at a distal part of the brain where you want those new cells to grow out to. You may want to be in, and that's one of the really promising features of focused ultrasound, is that you can target different parts of the brain uniquely to deliver drugs to those areas. So in the long run, it would be fantastic if a patient came in, we'd give them the initial gene therapy, they get some growth factors a week later to help. Not only have you rescued or down-regulated the amount of pathology, but now you've up-regulated the amount of growth factor that is involved in that. So those cells start to grow, and then 5 weeks later, as those cells are growing, but not knowing where to project to, you now deliver another stimulus or another vector to some other part of the brain and says, here, come over here. And, this is all science fiction right now, but we don't know where gene therapy is going to be. The vectors we use today are not going to be used in 5 years. They'll be ones that are much more specific, much more targeted. So I think there's, from a technical standpoint, gene therapy is very, very rapidly evolving. I think the drug delivery aspect for using focused ultrasound will become more and more efficient. So currently, the focused ultrasound transducer we use is a single transducer when we use in the animal models. The versions that are being developed for human treatment will have over 1,000 separate transducers for the sonication. That means there are sonications coming from different angles that you could have a very, very much of a focal point. And so as we get more and more precise, you'll find that the regions of the brain that you can target are also more and more precise. And this is a really exciting time for all of that work. So I'm happy to be a part of it, just a little part of it.
This has been incredible. Thank you.
No, you're welcome.
Today I'd like to discuss a paper entitled Robust Production of Uniform Human Cerebral Organoids from Pluripotent Stem Cells. I'd like to begin by answering the question, what is an organoid? Organoids are simplified organs that model three-dimensional in vivo activity. They're generated by growing stem cells in media, which allows for proliferation, migration, and differentiation into various cell types of a particular organ. The cells are then able to interact in a dynamic way rather than in a two-dimensional plane. This allows for cerebral organoids to be a more versatile alternative to animal models or two-dimensional cell culture in neuroscience research. Human cerebral organoids can be used to model development, they can be used to model disease progression, or the effects of drugs or therapies. This tool provides species-specific insight into molecular pathways because human-induced pluripotent stem cells are being used. And this is also relevant when studying context-dependent pathologies for a particular person. There are existing methods of generating organoids, and I'd like to touch on two of them. The direct patterning approach by the SSI group involves the use of small molecule regulators and growth factors to direct organoid development. This method involves more intervention on the researcher's part, as there's manipulation of EGF and FGF levels throughout the process, and essentially there's just additional steps taken during development to direct the organoid in a particular pattern of growth. Lancaster et al. Have developed a method that generates self-patterned whole brain organoids that effectively model brain tissue and microcephaly, and this method was modified in the paper I'm discussing to increase the utility of organoids as a tool. So the existing protocols demonstrate variability between individual organoids and variability between batches, which consequently hinders their application potential as a model. Therefore, the key lies in refining existing methods of generating organoids in order to produce uniform cerebral organoids. The authors of the paper I'm discussing were able to generate organoids that accurately modeled cell type composition and morphology of the human forebrain. Some of the key factors that contributed to this were the removal of rock eye, altering basic FGF levels, and the removal of matrigel following embryoid body development. Early intervention was necessary to produce consistent self-pattern organoids that develop correctly. Well shape and coding are also relevant factors that I will touch on later. So this protocol aimed to standardize the production of human cerebral organoids, and in doing so has become an ideal tool for modeling neurological disorders and for drug testing. The figure below shows bright field images of a developing cerebral organoid across a period of about 24 weeks. Some notes on the materials and methods. H9 female embryonic stem cells were used to generate embryoid bodies first, and then the results were validated using H1 male embryonic stem cells. Next, 4 different types of well plates were used, and embryoid bodies were grown in either uncoated U-bottom plates or coated U-bottom, V-bottom, or agro-well plates. The coating just prevented embryoid body binding to the well. Basic FGF and ROC-I levels were manipulated between 0 to 4 weeks of development. Basic FGF concentration was lowered to 2 nanograms per milliliter to encourage neural induction, and ROC-I, which is a factor that promotes pluripotency, was removed to encourage differentiation. Immunofluorescence microscopy was used to localize markers in organoids. Single-cell RNA sequencing was used to track cell type composition. And lastly, electrophysiological recordings were taken to verify the presence of electrical activity in neurons. And the images on the right here show progression of electrical activity from a developing organoid up top to a mature organoid on the bottom. There are 3 major results of the study that I'd like to cover. First, the researchers found that the V-bottom well plates with the non-binding coating produce embryoid bodies with the most uniform density and the most uniform diameter. This is demonstrated in C and D of figure 1 in the paper. As you can see, the rightmost column of figure C shows nearly 100% of the embryoid bodies grown in the coated V-bottom plates had uniform density, and the rightmost part of figure D shows uniform diameter among embryoid bodies grown in V-bottom plates as well. The coated egg-roll plates are second to the V-bottom plates in uniformity, but the separation process of the embryoid bodies just makes the use of them far too tedious to be practical. And lastly, figure E shows how basic FGF concentration affects the radialization of neuroepithelium. 2 nanograms per milliliter was found to be the ideal concentration 2 weeks after initial organoid development. The radialization of neuroepithelium is necessary for proper self-patterned organoid growth. Therefore, the changes in basic FGF concentration were also necessary. The next major result relates to the localization of markers present at stages of cortical development. Markers were co-stained with DAPI and images were produced using immunofluorescence microscopy. The figure shown here on the left demonstrates that CTIP2, which is the deep cortical layer marker, is expressed before SATB2, which is the upper cortical layer marker. And during the 4 to 12 week period, both factors surround the SOX2 ventricular zone, which is a marker for radioglia. This area is illustrated using dotted lines, as you can see where SOX2 is most concentrated, and the borders between the layers becomes less apparent later in development. The organization of layers mimics that seen in vivo cortical development. The last major results support that individual organoids had cell diversity and there was uniformity of organoids across batches. The figure on the left is a uniform manifold approximation and projection plot of the 3 batches of organoids. So cells are plotted and their clustering pattern determines their cell type. And diversity among cell types is more apparent later in organoid development, but in this figure, which is based on organoids at 12 weeks, it notes presence of neurons, radioglial cells, and intermediate progenitor cells. Unlike UMAP plots of more mature neurons, this figure does not show presence of interneurons. The figure on the right depicts UMAP plots for 2 individual organoids across each of the 3 batches, and you'll notice that the plots follow the shape of the figure on the left. It captures the uniformity between individuals of the same batch and across batches. So the conclusions of this study: this model is highly reproducible, robust, and effectively models a human forebrain. This is supported by the consistent embryoid body diameter and density, localization of markers, and the UMAP plots. Early manipulation of conditions was critical in order to self-direct the organoid growth. Examples of this include removal of ROC-I, altering basic FGF, and the removal of matrigel. Excess matrigel was found to prevent ideal morphology during development, so that's why it was critical. These methods generated uniform cerebral organoids for weeks across the development period. And the standardization of this tool improves its application in research and can improve our understanding of critical development pathways in the human cortex, as well as provide therapies for existing disorders. The application of the tool developed in the study is the future of modeling disease in neuroscience. The increasing prevalence of neurodegenerative diseases like Alzheimer's mark a demand for research into therapies and treatments for these disorders. Studies may focus on genetic risk factors and trace development using induced pluripotent stem cells of high risk individuals. Risk factors can include particular mutations. So, for example, Fan et al. Modeled Alzheimer's using mice induced pluripotent stem cells to derive organoids with APP and PS1 mutations. They found an increase in astrocytes and glutamatergic neurons and a decrease in GABAergic neurons. This can be expanded upon by using human organoids to model the progression of Alzheimer's in humans. Organoids can also be used as a tool to uncover complex developmental pathways in autism spectrum disorder. Mariani et al. Developed organoids using induced pluripotent stem cells from individuals with idiopathic autism and explored the overproduction of inhibitory neurons caused by FOXP1, for example, GABAergic neurons. Despite the advances in our understanding of autism spectrum disorder in the past 2 to 3 decades, much is to be uncovered about it at a molecular level. I hope that we can continue to use organoids to do so. Lastly, I'd like to touch on the therapeutic potential of organoids in spinal cord injury or even blindness. This type of application was made aware to me by Dr. Adesano during our interview, and it relies on encouraging the cerebral organoids to grow out into the damaged areas and innervate them. The repaired spinal cord can then be studied by adding factors to redirect growth or differentiation. I genuinely hope I was able to convey the capacity for research that this protocol has and how amazing the work that was done in this paper is. Thank you for listening.
Today, I'll be talking about this paper entitled Gluten-Related Illnesses and Severe Mental Disorders. This is a review paper from the Roger S. McIntyre lab. Now, to first understand this association, we need to talk about what gluten is and what pathologies are associated with it. Gluten is a protein that's found in many processed foods, including in wheat, barley, and rye. When digested, it's broken down into gliadin and glutenin. Gliadin will come up later in the study. Celiac disease is probably the most common disorder associated with gluten. It's an autoimmune disorder where antibodies against gluten and transglutaminase are overproduced. Transglutaminase is an enzyme which deamidates gluten. This deamidation of gluten makes it more likely to stimulate the immune system. However, overall, this overproduction of antibodies leads to negative consequences such as gut damage, and gut damage leads to malabsorption of nutrients. Gluten sensitivity, on the other hand, is only diagnosed when celiac disease is ruled out, so there is no overproduction of antibodies and no gut damage present. Due to this, some studies have posited that gluten sensitivities are as a result of the nocebo effect, where inert substances, such as gluten in this case, cause a negative effect in the person. The treatment of celiac disease at the moment is a lifelong gluten-free diet. Interestingly, some preliminary studies have shown that there may be an association between gluten-related illnesses such as celiac disease and psychotic and mood disorders such as schizophrenia, major depressive disorder, and bipolar disorder. One study has shown that celiac disease diagnosis actually precedes the onset of schizophrenia symptoms. Another study showed, on the contrary, that celiac disease is significantly associated with non-schizophrenic, non-affective psychosis. Another study actually showed that there is no association between celiac disease and schizophrenia. So this shows that there's a history of various different results in studies showing various different associations between celiac disease and schizophrenia. Major depressive disorder and bipolar disorder have also been shown to be associated with subsequent development of celiac disease. And so this paper is trying to answer the question, is there really an association between celiac disease and these psychotic and mood disorders? To answer this question, the authors conducted a literature review. They searched databases using keywords such as gluten and other keywords related to psychotic and mood disorders. Overall, this process involved 4 steps. The first step was identification of articles to be considered. This involved searching many online databases, such as PubMed, using the aforementioned keywords. The next step, a screening phase, assessed if there were any duplicates of any articles. However, no duplicates were found. The third step is an eligibility step. This step involved reading the title and abstracts of each article, and then assessing them for eligibility. Overall, 83 articles were excluded in this step, and 38 articles remain. Finally, the fourth step was the inclusion step. Here, the articles were fully read through and fully screened to see if the information would be helpful to the literature review. Overall, 14 articles were excluded in this step, and 24 articles were used for this literature review. I've grouped the results of the literature review into different categories, the first of which is genetics. Overall, this review conducted by the McIntyre lab found specific genetic associations between schizophrenia and celiac disease. For example, the 6p23 p22.3 locus that is found mutated in celiac disease is very close to the dysbindin locus, which mutations are associated with schizophrenia. The review article also looked at a gene called myo9b, which is involved in epithelial barrier function. Mutations in this gene are actually also associated with celiac disease. However, they found that one study had shown that SNPs in this gene are associated with schizophrenia, but another study did not reproduce this association with schizophrenia. So once again here, we see some association with schizophrenia with the 6p23 p22.3 locus and possibly the myo9b locus. However, this was not reproduced in all studies. The next category of results I'll be going through are biomarkers. One study showed that anti-gliadin IgA and anti-tissue transglutaminase antibodies are actually elevated in those with schizophrenia. Recall that gliadin and transglutaminase are two proteins that are heavily involved in the celiac disease pathogenesis. Gliadin is the breakdown product of gluten after digestion, and transglutaminase is that enzyme in which many antibodies are produced during celiac disease. And so there's this interesting link between celiac disease and schizophrenia here. The authors posited that there could be gut damage occurring in schizophrenia similar to in celiac disease. However, further investigation showed that these schizophrenia patients actually had high anti-tissue transglutaminase 6, which is an antibody released in the brain. And so they posited that this could be possibly due to an interaction between gluten and the blood-brain barrier. Another study showed that the schizophrenia reaction to gluten may not be the same reaction seen in celiac disease. These reactions have similar immunological abnormalities, but the specific antibodies against gluten are different in schizophrenia. So once again, these biomarkers results show discrepancies between results across different labs. This next category of results will discuss the links between major depressive disorder, or MDD, and gluten-related illnesses. Overall, studies have shown that MDD is associated with elevated levels of pro-inflammatory cytokines. In celiac disease, T cells activate interferon gamma in reaction to gluten, and this has interesting implications in terms of the serotonin production pathway. In non-hepatic tissue, tryptophan is metabolized by an enzyme called IDO. Metabolism of tryptophan decreases the availability of it, and so there can be less production of serotonin. IDO is interestingly activated by IFN gamma. So overall, in celiac disease, we may see an increased activation of IFN gamma by T cells, and that in turn leads to increased activation of IDO. This decreases the availability of tryptophan as it's getting metabolized, and overall decreases the availability of serotonin, which has implications for major depressive disorder. final category of results I'll be discussing are the links between bipolar disorder and gluten-related illnesses. Bipolar disorder has also been associated with inflammatory response imbalances. Specifically, one study showed that people with bipolar disorder have high anti-gliadin IgG antibodies and also deamidated gliadin. Recall that deamidation is the reaction that transglutaminase does to make gluten stimulate the immune system more. And so here we see an interesting similar pathology in bipolar disorder and celiac disease. Another study found that those with mania have high anti-gliadin IgG, but not other markers for celiac disease. In that study, after a 6-month follow-up, they found that the antibody levels actually were not significantly different anymore from the controls. However, those who did have higher IgG at that point at the 6-month follow-up were associated with rehospitalization during that 6-month period. This once again shows a possible link between celiac disease and bipolar disorder. However, the results overall were insignificant. There are still some interesting tidbits of information that could be found there. Overall, the results showed that those with celiac disease may indeed actually have a slightly higher risk for schizophrenia, major depressive disorder, and bipolar disorder. There was also some data, although not conclusive, which showed that those with schizophrenia, major depressive disorder, or bipolar disorder may also have an abnormal response to gluten that is similar to that in celiac disease. However, overall, there isn't enough evidence to prescribe that lifelong gluten-free diet to people with psychotic and mood disorders. In the future, it could be interesting to explore the possible bidirectionality of the relationship between celiac disease and psychotic and mood disorders. This literature review pointed out that there may be possible that someone with celiac disease is more likely to get diagnosed with schizophrenia, major depressive disorder, or bipolar disorder. However, in that light, is it also possible that someone with schizophrenia, major depressive disorder, and bipolar disorder is also more likely to be diagnosed with celiac disease? Some results in the literature review did point to this. However, this has not really been discussed or explored very much in past literature. Another future direction for authors could be to look at whether gluten sensitivity could be controlled by targeting the nocebo effect. If you recall earlier, gluten sensitivity is diagnosed by exclusion, and so there is no gut damage and no increased antibody production as seen in celiac disease. However, those with gluten sensitivity still see some symptoms that are seen in celiac disease, such as overall bloating when eating items with gluten. It is known that the dorsolateral prefrontal cortex can be targeted to control the placebo effect. And so this may be a possible therapeutic route for those with gluten sensitivity, in that by targeting the dorsolateral prefrontal cortex, we could help decrease the symptoms of gluten sensitivity. Thanks for listening.
Okay, hello, my name is Ritaj. This is my final presentation for HMB 420. I was interested in studying the neurobiological underpinnings of anorexia nervosa, and so I reached out to a lab in New York that published this article last year called Voluntary Wheel Running Exercise Evoked by Food Restriction Stress Exacerbates Weight Loss of Adolescent Female Rats, but also promotes resilience by enhancing GABAergic inhibition of pyramidal neurons in the dorsal hippocampus. So quick overview of anorexia nervosa. It's the most lethal psychiatric disorder characterized by restrictive caloric intake and usually associated with extreme exercise to further reduce weight. The adolescent female population is the most vulnerable, and a multitude of biological, social, and cultural factors are thought to underlie the disease. Research on the neurobiological underpinnings is scarce, but neuroscience is an invaluable tool in providing us with information on anorexia nervosa. It can answer questions like, what do we know about the changes that the brain undergoes during AN, and what are its causes, disease profile, and symptoms, as well as if there are pharmacological treatments that can be used to target the brain. So this is the lab I reached out to. Their paper is critical to this field because it's presenting a plasticity-mediated molecular mechanism for resilience in anorexia nervosa and showing what happens in the brain during AN using a mouse model for the disorder. And so the researchers are evaluating the role of inhibitory GABA receptors and GABAergic synapses in the hippocampi of these animals. So they wanted to know what changes the mouse brain undergoes, what the role of GABA is in these changes, and whether the full maladaptive spectrum, that is food restriction and excessive exercise, is required to see these changes and see the resilience, or if only certain elements like food restriction or excessive exercise are required. And so they were using female Sprague-Dawley mice divided into 4 conditions, and these are all adolescent female mice housed in isolation. They are looking at the CA1 pyramidal cells of the dorsal hippocampus involved in the stress response, as we'll see in just a minute, and at the GABA currents via whole cell patch clamp technique. Electron microscopy is used to analyze the structure of tissues, and ICC, or immunocytochemistry, is used to find and bind proteins of interest to determine GABAergic synapses, which are profiled via a GAD marker and identified using the rabbit anti-GAD antibody. The ABA mouse model is the mouse model of anorexia nervosa, and so it's induced in about 8 days. For the first 4 days, the mice are fed and allowed to exercise as they like, and on the 4th day, food restriction begins. The food restriction is very stressful for the animals, and so they'll engage in foraging behavior, running, as they would in the wild, but in a cage this is maladaptive because they're not getting anywhere by running on the running wheel and just using this as a mechanism to curb their anxiety. At some point in the next 24 hours, the mouse will begin to restrict its food intake even when it's offered food. And so it will voluntarily run and it will voluntarily restrict food. And this culmination of food restriction and excessive exercise is modeling anorexia nervosa. And so in this experiment, we have this activity-based anorexia mouse, which I'll just call the anorexic mouse from now, and the control, which is given food but no running wheel, the food restriction condition, which is also food restricted but given no running wheel, and the exercise condition, which is not food restricted but provided with a running wheel. And so the experiment runs for about 8 days, and on the 4th day, we start restricting food for ABA and food restriction. And throughout the next 4 days, animal weight and wheel running activity is measured. And after about 8 days, the mice are decapitated and the brains are extracted for analysis. So what they found was that food restriction paired with excessive exercise causes hyperactivity in mice and exacerbates their weight loss. So you can see that on the graph on the left here, body weight is being mapped against experimental days, and body weight is not significantly different between the mice until about day 4 or 5 when we start restricting food. And you can see that the body weight of the anorexic mouse group significantly declines in comparison to just the food restriction or just the exercise condition, the latter of which actually gains weight. On the right, you can see that 24-hour activity in kilometers is being mapped against experimental days. And for the mice that are provided with a running wheel, so the anorexic mouse group and the exercise group, at about day 4 or 5, you can see that the anorexic group is engaging in a lot more exercise, at one point hitting 7 kilometers a day. When you think about how small that running wheel is, this is significant because it shows that the mouse is incredibly stressed, and in comparison to the exercise condition only is running a lot more. And so this excessive exercise in combination with food restriction decreases the activity-based anorexia's body weight by a lot, significantly more than either condition alone. Whole cell patch clamp technique is used to show that GABA currents are higher in the anorexic mice than they are in the control. So that's that graph on the left here. You can see that the current measured in picoamps is a lot higher for the anorexic mice than they are for the controls. And this is kind of strange because if you look at graph B in the middle here, the GABA response should decline after development, because we know that GABA is very important in early development, but not so much so as the mice grow. And so this lends evidence to the idea that the brain is undergoing this increased inhibition in the anorexic condition that it's not engaging in for the control or for the food restriction and exercise condition alone, as you can see on graph C. So the anorexic mice have a greater response to GABA than any one of the conditions alone. This is your EMICC, and so electron microscopy and immunocytochemistry is showing here that these black arrows, I don't know how clear this is, but these black arrows are showing you wherever they're finding GAD, and GAD is a GABAergic synapse marker, and you can see in the anorexic mouse, there is a lot of GAD, and the synapse is heavily populated, so the synapse has clearly undergone changes that the control has not, and this is the control. And you can see that it has less of these GAD markers. And so when it comes to excitatory neurons in the dorsal hippocampus, these are the CA1 neurons, they are showing elevated GABA markers in the anorexic condition. Okay, and this last result I found the most interesting. GABAergic neurons are actually unsilenced via changes in the GABA receptor subunit, and ultimately there is more GABA signaling in the anorexic mice than there is in the control. So you can see the control here is silent, and we don't have a lot of receptors as compared to the activity-based anorexia. These GABAergic neurons upregulate their GABA receptors and become unsilenced, so increased inhibition in the hippocampus. And that's also compared to food restriction and exercise. You can see exercise is pretty much silent, just like the control, whereas food restriction has a few more GABA receptors, but not as many as the activity-based anorexia mouse. And so this GABA receptor subunit, the mechanism is essentially that the GABA receptor subunit alpha-4, it undergoes a conformational change and turns into alpha-1, and this causes the translocation of extra GABA receptors from the extrasynaptic site to the synapse. So you start to have GABAergic neurons that are active and unsilenced in the anorexic mice. So why is all of this happening? The brain is essentially upregulating GABA receptors and GABAergic synapse signaling in the dorsal hippocampus to compensate for the increased excitation caused by food restriction stress. So food restriction is a very stressful condition for the mouse, and the excessive exercise means that there is a lot of excitation in the hippocampus. And so the brain is basically undergoing this compensatory mechanism by upregulating inhibition via GABA to kind of downregulate that stress response. And so GABA is acting as an inhibitory, so the GABAergic synapses and the increased GABA signaling is inhibitory and attempting to decrease excitation. And it seems that the increasing GABAergic plasticity is resulting in the increased GABA response, because the anorexic mice show a greater GABA current and are more responsive to GABA than the controls. And at least based on this evidence, it seems as if food restriction and excessive exercise are both necessary in order to see the resulting changes, because in either condition alone, so just food restriction or just exercise, you're not seeing the increased GABA current, the upregulated GABA receptors, or the extent of weight loss and hyperactivity. And so the full maladaptive conditions are required to see both the pathology and the resulting GABAergic plasticity. So I did speak with the researchers. I had a lot of questions, but I've just inserted a 2-minute clip here of what I thought was the most interesting notes that she had.
And that's a really interesting question. And that experiment has not been done. I tried to do that myself, of injecting a GABA receptor agonist into the hippocampus, and I was not very successful with that, because what I had to do was to implant pipettes, like cannula, into the hippocampus, and to deliver the drug slowly and chronically over many hours. And that sheer implantation of the cannula, it caused a lot of destruction of the hippocampus.
Oh, wow.
And the destruction was so bad that it sort of obscured any pharmacological or therapeutic effect, if there were any.
Oh, wow.
So I went step backwards, and what I did was, instead of trying to directly infuse GABA into the hippocampus, I chose to use a drug which I could inject into the tummy area. That's called intraperitoneal, to provide systemic, that means the whole body, whole brain, whole body delivery of GABA, giving up on the idea of what is the role of GABA specifically in the hippocampus. And when I tried that treatment, I was able to see a reduction of wheel running. So that was very helpful to me, because I believe that the animals that run more on the wheel are the ones that are anxious.
Wow.
And so they ran less, which was very helpful, because I think that means they were anxious less, and that's why they didn't run as much.
Okay, so yeah, I thought that was really interesting that she had done the experiment that I had questions about, and had found those results. Future directions and critical analysis. I have three major points that I want to address here. The first being the role of isolation stress in the experiment. And this is kind of a critical look at the mouse model itself. So we know that social isolation is a mechanism by which we can induce depressive-like symptoms in mice. And so it's important to determine the extent to which food restriction and the weight loss and the hyperactivity could be as a result of social isolation, and not just the actual mouse model. So is the mouse running excessively and choosing to restrict food because it is also socially isolated, and not just because we are restricting its food intake? In fact, other studies in the field have shown that mice housed alone have elevated corticosterone levels, and that when provided with a running wheel, those measures of corticosterone, so that's our marker for stress, are even higher as compared to controls. So a way in which this can be addressed in future studies is to develop another condition with activity-based anorexia mice, but ones that are housed together, and determine if these mice show the same response as the activity-based anorexia mice housed alone, or if perhaps having a social support or another mouse with them would decrease those symptoms and decrease that pathology. There's also something to be said about the effect on learning and memory in the activity-based anorexia model that has gained resilience. So when we dampen excitability of the dorsal hippocampus region by upregulating this inhibition through the GABAergic synapses and the GABA receptors, what are we doing to other functions of the hippocampus, like mood, emotion, memory, and learning? And this is important to consider when we try to apply these results to the human population. So studies in the field have found that mice lacking a vital subunit of the GABA receptor for the hippocampus actually perform better on memory tasks, and so learning and anxiety tasks, And so in the wild type, the presence of the fully functioning GABA receptor is decreasing the performance in these learning-based tasks. And so there's something to be said about upregulating inhibition in the hippocampus and what it's doing to the other functions of the hippocampus. So future studies could look at learning and memory in the activity-based anorexia mice, and see, are they scoring higher or lower on these measures, because we have this increased inhibition on the hippocampus. And finally, future studies should consider the role of relapse. So that's critical because in the human population, anorexia nervosa has a very high relapse rate. And so further experiments could induce multiple activity-based anorexia sessions. So the mice that gained resilience during the first session, so resilience as is measured by the upregulation in GABA and the increased weight, if they can stand up to further inductions. And so important questions here would be if there's a limit to GABAergic upregulation, if resilience gained during the first or second induction hold up against further induction, and if those ABA mice that have gained resilience can be made to relapse with further food restriction. That is everything I have for today. I think I'm a little bit over the time, so I will stop now, but thank you so much.
Hi there, my name is Paulina Rubitska, and I will be focusing on research conducted by the Holmes Lab at the University of Toronto, which is detailed in a scientific article called Contrasting Effects of Opposite versus Same-Sex Housing on Hormones, Behavior, and Neurogenesis in a Eusocial Animal. So, to give a little background, in mammals, including humans, some regions of the brain retain the ability to produce new neurons throughout life, a process called adult neurogenesis. The goal of this research is to determine how social status controls this process in the mammalian brain and how these new neurons might contribute to status differences in behavior. The Holmes Lab uses the naked mole rat to study this question because they exhibit the most rigidly organized social and reproductive hierarchy among mammals. They're eusocial, which is essentially an extreme form of cooperative breeding. And a thing to note is that within the colony, there is a queen, which is the only reproductive female, and there are 1 to 3 reproductive males. Everybody else is unable to reproduce, and they are called the subordinates. But if a subordinate is removed and paired with an opposite sex individual, they are able to reproduce. Also, given that these animals are very long-lived, up to 30 years in captivity, there's great potential for plasticity mediated by the social environment. Also, previous work has found that breeders have significantly decreased neurogenesis in the dentate gyrus of the hippocampus compared to subordinates of either sex, and this is particularly driven by low neurogenesis in queens. So in this study, the subordinates were removed from their home colony and paired with either an opposite sex or same-sex animal for 1 month, and the behavior was recorded. Interestingly, they found that the opposite sex paired animals showed increased neurogenesis relative to subordinate controls, while same-sex paired animals did not, although it is important to consider that same-sex paired animals showed increased aggressive behavior. Collectively, this data suggests that neurogenesis is highly fluid as animals transition status, and this is likely based on the specifics of their social and endocrine environment. So why is this paper important, and what unanswered questions did it resolve? Well, this paper is important for a number of reasons. First of all, it clarifies the mechanisms that underlie competitive interactions' effect on experience-induced plasticity, specifically neurogenesis and HPA activation. Also, previous work has only looked at the competition for breeding status within colonies of naked mole rats, and not in a colony-founding context. And other papers have not assessed the neural and endocrine mechanisms involved, which is really important. So ultimately, this paper's results improve understanding of the effects of social stress on neuroplasticity and behavior. Okay, let's look at the methods now. So the young aged adults were randomly assigned to one of the 3 groups, either colony house subordinates, opposite sex pair house subordinates, and same-sex pair house subordinates. These animals were maintained out of colony and left to interact with their partners for 4 weeks prior to tissue collection. After sacrificing the animals, immature neurons and proliferating cells were labeled respectively using DCX and Ki67. DCX and Ki67 immunoreactivity were evaluated throughout the rostral caudal parts of the dentate gyrus and the basolateral amygdala. Additionally, to assess hormonal levels, blood was collected at the time of euthanasia, after which testosterone, estradiol, progesterone, and cortisol levels were measured by enzyme-linked immunosorbent assay, or short form ELISA. And now for the results. And overall, the results suggest that cooperative and competitive housing differentially modulate behavior, stress physiology, and limbic plasticity in this eusocial animal. After chronic pairing, opposite sex animals exhibit affiliated behavior, for example, huddling, confirming that opposite sex housing facilitates cooperative interaction. In turn, hippocampal DCX and Ki67 expression are enhanced in these animals relative to subordinates in colony. They also saw that there was an increase in DCX expression in the basolateral amygdala only in males. And this increase is triggered by opposite sex housing, suggesting that pairing with a female enhances amygdala neurogenesis for males. Lastly, the baseline glucocorticoid levels were unaffected by this opposite sex pairing, but augmented above colony levels in same-sex animals, as we'll see in the next slide. And with same-sex animals, there was no neurogenesis spike seen, as in no DCX and Ki67 expression within the hippocampus. Same-sex pairing also appears to facilitate agonistic interaction, with same-sex animals expressing more aggressive behavior, such as shoving, after extended contact. They also saw that female, but not male, same-sex animals exhibited enhanced DCX expression in the basolateral amygdala relative to subordinates in colony. This means that the females had enhanced amygdalar neurogenesis. These differences, so aggressive behavior and amygdala difference, cannot be attributed to classic male competition, as aggression was limited to female-female pairs. Also, interestingly, enhanced basolateral amygdala cell survival was associated with an eventual increase in antisocial behavior amongst the female rats. Lastly, glucocorticoid levels were above colony levels for both sex pairings. In conclusion, the work from the study is important as it enhances the understanding of the effects of social stress on behavior and neuroplasticity. Results verify that social stress, specifically female-based competition, exerts contrasting effects on hippocampal and amygdalar neurogenesis. This research is crucial, as this naturalistic conflict among group living animals very closely mirrors human relational stress.
Culfurum, Y-U-R-I-Y-B-A-L-K-O. Culfurum, Y-U-R-I-Y-B-A-L-K-O.
May alter any communication between neural systems and modify emotional responding. So this enhanced basolateral amygdala cell survival is connected to an increased antisocial behavior among females, consistent with human-focused studies where patients with stress-related disorders experience increased vascularity and varied resting state connectivity, specifically in the basolateral amygdala. So this research is crucial, as past work has found that women are more responsive to social stimuli than men and are more vulnerable to developing affective disorders. So this work can ultimately help improve understanding of affective disorders by detailing the neural mechanisms for stress-induced change in behavior. So I think there are about three main directions that can be taken in the future to understand and advance the knowledge of this topic. So first I want to focus on the markers that were chosen in this study and how they can be improved. So in this study, researchers chose DCX and Ki67 as their endogenous markers of proliferation and neurogenesis. So this allows the researchers to get a more broad view of neural plasticity because the proteins are expressed for a longer period of time, particularly DCX. Another approach to neurogenesis is to use exogenous markers, for example, Burju or Edu, which essentially birth date cells at a more precise time point. And this is a very powerful approach, but requires more knowledge about how the cell proliferation, survival, differentiation cycle works in a species in order to design experiments well. So as mentioned, moving towards a more precise manipulation approach using Burju and Edu would help get more insight into how social manipulations influence different aspects of the neurogenesis process. Next, I want to focus on the patterns of neural plasticity that underlie same-sex hostility in relationships. So as we observed in this study, there was an increase in female-to-female hostility in the same-sex coupling. But in past studies, we focus on mostly male-to-male aggression. And with that, there's, I want to say, a dogma that social hierarchies matter for males and not for females. And, this is absurd, as hierarchies are hugely important for females, and we see that even with humans, right? Inevitably, there must be some differences, though, in terms of these patterns of neuroplasticity. But what are they? And how similar are they for both genders? And how can we explore that further? So we can look at other species, such as hyenas and meerkats, as both species are very social and live within strong female dominant hierarchies. So comparing them to the naked mole rat would allow us to see how conserved some of these effects are. And lastly, the study only looked at neurogenesis occurring within the dentate gyrus of the hippocampus. But we know that the olfactory bulb is also a target for that. So another area to focus on would be the olfactory bulb. for example, through ablation, where neurogenesis continues through adulthood. And as Dr. Holmes mentioned in our interview, her hypothesis is that ablation of neurogenesis in this subventricular zone or a factory bulb circuit would impair social recognition of conspecifics, which would impair their ability to maintain the social hierarchy. So that's a really interesting direction to also pursue. I think targeting a completely different brain area, but also which is responsible for adult neurogenesis. So overall, it's not just essential to study female-dominated species, as we can see. It's important to study females in general. And females do form and maintain these social hierarchies, even mice, for example, and obviously in humans. But they're different from the traditional hierarchies that are studied in males. For example, aggression can be different, what resources are pursued, etc. So I think overall, there should be a much more rigorous approach to female studies, as this study evidently shows that there's a lot of potential there and a lot to learn, especially in relation to stress-related disorders. So last thing I want to say is thank you. Thank you for listening and dedicating your time. I hope you learned something and you enjoyed the presentation.